soon as Suzanne, let's go on with Suzanne's talk, and Jonathan, you can ask your question later.
Okay.
Start when you're ready.
Great. So I am going to do an introduction, and then Bob Hitzemann, my collaborator, is going to talk about some of the genetics here. We are also looking at delay discounting. Like keto, we use a slightly different paradigm in which individual rats choose between sucrose rewards on a trial-by-trial basis. And the size of the rewards are just based on the choices. So somewhat different paradigm. In our UO1, we are doing a lot of behavioral phenotyping. Definitely resonate with the statement about it's hard to keep people employed and doing all this phenotyping because it's such hard work. The genotyping of the individuals gets done at UCSD. And then we have RNA Seq of 3 particular brain regions, the prelimbic cortex, nucleus accumbens core, and BLA. And we are also engaged in creating some selected lines, and that's ongoing. So at this point, we have 655 animals that have finished the phenotyping, and they look really similar here. We haven't got as many animals as keto has, but right now we see no sex differences at all. And you can see that in terms of the gradient. You can also see that in terms of the area under the curve measure. Very unfortunate, but the data are the data. I want to show you that there are lots of individual differences. So what becomes important is trying to quantify those, and a paper that I have under review looks at different quantification methods, and so hopefully that will come out soon. An interesting way to try and quantify where we do actually see sex differences is using the quasi-hyperbolic model that's often used with people. I'm unaware that anybody has actually used it with animals other than our lab. And so we're excited about quantification, but I know you all are going to be much more excited about genetics. And so I am going to turn it over to Bob Hitzemann. I'm going to control the slides, and he's going to tell you about some of our RNA Seq findings. Bob?
Yep, I'm here. Everybody hear me?
Yeah.
I can hear you.
Good. Okay. So this first slide here is an old slide. This was the genotyping that Abe and his lab did on the first about 400 animals. The SNP, unlike Olivier's data, the SNP heritability here is very low, and that's across all the different phenotypes that can be extracted from the data. So this is, I think, an example of where there's no large gene effects. If we wanted to find the Manhattan plat, I'm not going to show because there's nothing there. So again, the sample sizes are small. There's another 200 animals that are out there that went through the same procedure, and then we'll have those selected lines, which Suzanne can talk about in the question and answer period if anybody's interested. Next slide, Suzanne. So the RNA Seq data, we've taken the first 200 animals that were phenotyped, and we've done RNA Seq on them. This is a paper. This diagram here is from a paper by Bailey. It looks at some different cost-related phenotypes, and as you can kind of quickly see, many of the regions are exactly the same, depending on regardless of the phenotype. We're looking at C here, and we've looked at 3 brain regions: the basolateral amygdala, the core of the accumbens, and the prelimbic cortex. And we've collected data now in 100 males and 100 females from the first 2 shipments of animals that came from Wake Forest. Next slide. So the gene expression data, like many others that have talked today, we're about a year or a year and a half behind in collecting this data. It's largely pandemic related. We just simply couldn't get the samples through the sequencing center as fast as we liked, and there were delays and up and down the line. So, but anyway, we have all the data now. We finally got the last 300 samples about 3 weeks ago. So what I'm going to show today is some old and some new data. So the gene expression data is 25 million stranded paired end reads per sample, 3 brain regions, prelimbic cortex, Cummins core, and BLA. All the data has been collected now. The plan here generally is to create gene networks and determine which modules are most strongly associated with delayed discounting. And then at the hope we can integrate all the data that we have from the behavioral and expression and QTL data and identify the key hub nodes. One of the things we're looking for and working with Dan Monroe on is to identify the eQTLs in this data set. And Dan has just started working on that on the prelimbic cortex data. He hasn't seen all the data yet on the core and the BLA. Haven't been able to send it to him yet, but that's where we're going. So we're gonna look to see which of the key hub nodes are actually eQTLs, and with some idea that they might be causally related to delayed discounting. Next slide, Suzanne. Okay, so we're going to get to the sex differences that we see are not in the phenotype, but are in the gene expression data. So this is the BLA data. This is for log K, which is the same parameter that's used in many human studies. And the bottom line here is we could go through each of these things separately, but we'll save some time. The females and males are very different. And so the females are showing a fairly significant association with receptor signaling and the males and cell adhesion molecules. I will say with the males, we see a lot of signal with the cadherins and protocadherins. That's something that we found before in alcohol-related phenotypes, that these cell adhesion molecules tend to show up, and they tend to show sex differences as well. So males and females are very different. And what that actually means in terms of developing new therapies, we could discuss perhaps in the Q&A if you'd like. Next slide, Suzanne. So what you can do with this kind of data, and many people in the audience have done this before, you simply take the data, you form the network analysis. Here we're using the WGCNA, but we've done other network parameters, and they all comes about the same. And what we see is that in the females, there's a strong association with 2 of the modules in the WGCNA, the gray 60 and the light cyan. Again, we all know that the names of the modules have no meaning whatsoever. But the important thing here is to see we're associating the transcriptional data with the module eigengenes, and those associations are much stronger in the females than in the males. And so and we see that across all 3 brain regions. I'm not showing that data today, but it's the same in the core and in the prelimbic cortex. Next slide, Suzanne. So now what we've done, we've now got data with our new sample sizes, and we've increased the sample size, doubled it just recently. We've now find a new signal, and a signal is detected for the GO category of psyllium. There is no GO category for primary psyllium, surprisingly, but we assume there are no motile psyllium in the BLA. So this association was not detected for the log K phenotype, but rather for the traditional area under the curve. The psyllium signal was detected for both the male and female sample jointly, but the signal is largely derived from the female data, where it's much stronger. The psyllium signal is not found in the incumbent score or the prelimbic cortex. So why is this kind of important? Suzanne, the next slide. For those of you that don't know much about the primary psyllium, every cell has one, and it's basically like a little antenna that kind of kind of waves around out there a bit in the extracellular space, and it's very sensitive to changes in its environment. What's really interesting about the cilia, the primary cilia, is that there are some receptor proteins that are only found in the primary cilia, and they're listed here. So we see somatostatin 3 receptor, also the adenylocyclase 3 receptor, adenylocyclase 3, 5-HT6R. That's kind of an interesting one, since there's been a lot of interest in that receptor in modifying cognition, treatment of Alzheimer's disease, treatment of mood disorders. So they're both agonists and antagonists that can be used to modify that receptor, and that receptor is only, as we know, found in the primary cilia. So there's potentially new targets there for looking at how these receptors might modify delay discounting. I searched the literature, I couldn't find a thing that any drug that affect any of these enriched proteins actually has been tested in delay discounting, or even something more general, we'll just call impulsivity. That may be true. I just couldn't find it. So, but I think there's certainly potential there for some pharmacological manipulations to see what happens. I'm going to stop there. I'm done. As far as publications, I'll turn that back to Suzanne. But I think we're waiting to get the finish up this analysis now that we have our full RNA seq sample. We'll be working with Dan to get the data on the EQTLs. And so I suspect the publication on this will come out sometimes early next year. It will at least be submitted. And then, of course, our approach to actually confirming what we find in these first 200 animals is to use the selected lines that Suzanne's now making, and we'll actually be able to test those to see if we get the same differences between high and low discounting that we now see in this data we already have. So I'm done. Thank you.